Institute of Chemical Technology, Matunga, Mumbai, and Madam is going to present her topic on innovative formulation approaches to enhance livestock productivity. Today we have another speaker, Dr. P.S. Banerjee. He is working as station in charge, Eastern Region Station ICR IVRI, Kolkata Center, and he is going to present his talk on meat-borne parasitic zoonosis. So just we will wait for 2 minutes and then we will continue.
So today's seventh day of our training program, and now today we have very renowned n- Professor Madam Padmavi Devarajan Madam. She's uh, from the ICT, that is Institute of Chemical Technology, Matunga, Mumbai. She's a Dean Research and Innovation and a Professor in Pharmacy. Madam will speak today on innovative formulations approaches to enhance livestock productivity. And another speaker we have, that is Dr. P.S. Banerjee, is a station in charge, Eastern Region, Regional Station, ICR, IVRI, Kolkata, West Bengal. And he will speak on meat-borne parasitic zoonosis. So today the speakers will contribute their uh, expertise in this their expert area and their experiences during their talk. So I request Rajeshri Patil Madam to just introduce Madam Padmasri Devarajan to the participant. Dr. Patil Madam, please.
Thank you, Jhande sir. Very good morning to all of you. It's my pleasure to introduce today's eminent speaker, Professor Padmavi Devarajan, who is working as a Dean Research and Innovation, Professor in Pharmacy and former Head Department of Pharmaceutical Sciences and Technology at the Institute of Chemical Technology, Mumbai, India. Dr. Padma Devarajan Madam is also a member of the Board of Governors and head of the incubator at the Institute of Chemical Technology. She has many granted patents, has licensed technologies to industry and product commercialized in India and Europe. She is author of 2 books on targeted drug delivery published by Springer. She was actively associated with Controlled Release Society Inc. USA as board member, member on the board of the scientific advisors, chair of the Young Scientist Member Protege Committee and Chair of the Outstanding Paper Award Committee of the Journal of Drug Development and Translational Research. She is a member on the editorial board of the Asian Journal of Pharmaceutical Sciences and the European Journal of Drug Metabolism and Pharmacokinetics. Professor Devarajan Madam is a nominated fellow of the Maharashtra Academy of Sciences and life fellow of the Indian Chemical Society and president Society for Pharmaceutical Dissolution Science, SPDS. She is a recipient of several awards and recognitions for research and innovations, including the American Association of Indian Pharmaceutical Scientists Distinguished Educator and Research Award, the Vivid Lakshmi Audyogik Sansthan Vikas Kendra Award for Industrial Research, uh, the Association of Pharmaceutical Teacher of India Award for Research in Pharmaceutical Sciences, OPPI Scientist Award, and the Outstanding Woman Scientist Award of ICR, Government of India. So with such a with a very expert background and with a lot of contribution in the field of this drug delivery and all, we are happy to have today's eminent speaker, Dr. Padma Devarajan Madam. So now I request Padma Devarajan Madam to please deliver the speech. Thank you.
Thank you so much, Dr. Rajeshri. At the outset, I would like to thank all the organizers for giving me this opportunity, and particularly since the last 10 years, I have kind of been working extensively in the veterinary field. And that is thanks to a wonderful team I have met from the Bombay or Mumbai Veterinary College, starting with Dr. Samad, then Dr. Gaikwad, then Dr. Maji, and currently we have a joint project that is Dr. Rajshri Gandage and myself from ICMR. So this collaboration continues today academically, and it's continuing also on the business front with Dr. Samad. So with this, I'm really happy to have this opportunity to talk to a lot of veterinarians on an area probably which you would not focus on, which is, let me start sharing. Can you see the slide?
Yes, ma'am. Yes.
Innovative formulation approaches to enhance livestock productivity. Because whatever the veterinary people would work on would be what is available already in the market. And I have seen that while there's a lot of research in veterinary area with reference to nutrition, tackling infections, understanding and diagnosing infections, there is limited research in the formulation area. And I must acknowledge that whatever I would be presenting to you today is a result of very active, or rather very proactive collaboration of a team of veterinary scientists, other scientists, biological scientists, and with the ICT team, with our team that is technology-driven team. So with this today, I would be presenting to you some of the innovative formulation approaches that we have been working on, and which actually I realized when I saw the theme of the conference that it does relate directly to livestock productivity. And although I haven't put it here, also food safety comes into picture automatically, or rather like it's a ek pe ek free jo bolte, it's like that. So while we have been working on livestock productivity, we have also been addressing the safety. So livestock is something that we started focusing on because of the collaboration that was initiated. And we understood that India being an agricultural economy, livestock plays a very vital role. And in India particularly, we have livestock producers who are small or marginal holder farmers. And I think particularly in the dairy farming, they're about 80%. And India is among the largest producers of milk in the world. So one very important thing that I would like to stress on right now is that while we have been working on innovative formulations, our major focus has also been affordability, because the people we want to develop for are not just the rich dairy farms or the big dairy farm, but we want to support the smaller farmers and the marginal farm holders. So with this, what did we focus on? Now, we have been working a lot on infectious disease formulations for human use. And then when we got into the veterinary field, we realized that some of the concepts we were using there could be wonderfully adapted also for veterinary application. The challenge, when you see the kind of formulations I talk of, which are in the industry for humans, is very expensive. What we are doing is making them simple and making them affordable. So why did we choose infectious diseases? One, because we realized they are really rampant. Almost 50% of the population sometimes has a particular disease, and they are difficult to eradicate, particularly when the infections are intracellular. When you treat an animal, something that you don't look at in humans is the drug residues that could come in the products from animals like milk, meat, etc. They do deteriorate the health of the animal, and therefore there's complete reduction and sometimes loss of productivity. And an even major challenge is zoonosis, which I'm sure all of you understand, and I believe the next lecture is all about zoonosis. So these are some of the intractable veterinary infections that we were looking at: brucellosis, theileriosis. And if you can see, more than 25% of the livestock population could be at a particular point of time afflicted by these. And these include cattle, goat, horses, pigs, crossbred cows, and canines. And they're transmitted through bacteria and ticks. So what was our concept is to have something to treat the veterinary infection so that you do not have zoonosis and you make all these animals free of infection and the products that come out of animals safe for human use. The current solution, we were told, is slaughter. And in India, slaughter is not permitted. So people or farmers are forced to live with these animals, tend to these animals, which means they have the chance of falling sick themselves and also not earning from the animal while spending on them. So all this told us, yes, why not look at this area? And we had a very nice team that supported us in this. So now I'm going to present you, based on this brief background, about why we got into infectious disease treatment in the veterinary field. I'm going to present you 3 of the basic systems that we've done. And my request to the organizers is, when there is 10 minutes left, please signal to me so that accordingly, if I'm too long, I will wind up comfortably. So I'm going to look at one aspect, and that is targeted delivery. And this is very important because we will be targeting our system to the site of the infection and not just doing an administration where it goes all over the body. We are also looking at tailored nanosystems for targeting, which means we use very innovative technology to make nanosystems to behave in a particular way that we want. I will show you an example where we have tailored the shape of nanosystem. And we are also simultaneously addressing innovative technologies. So one of those would be an innovative nanotechnology system that I present, and another one would be for mastitis. So the 3 basic, let me say, target infections that we were looking at, one was Teniosis, where we have a success story, which a commercial product may be launched soon. Mastitis, our success story is already launched. I will show you some information. And the third is brucellosis and other intracellular infections. The drug we have chosen for is brucellosis, which could also work for ehrlichiosis. So the contents of my talk would basically be innovative nanotechnology, where I will be presenting to you solid lipid nanoparticles, which is a nano delivery system of this drug buparvacone, which I'm sure all of you are familiar with, used for this infection theileriosis. A tailored nano system, which is an asymmetric lipomer. This is again a nano system which is not round. Asymmetric because it is not round. And if you look at papers and you look at liposomes, solid lipid nanoparticles, you'll find a lot of pictures that show you round particles. Here we'd have particles that are not round, and therefore they work beautifully for spleen targeting. And the third is a very simple but interesting and innovative formulation, which is a plug forming tea dip. So this is drug free, very important. So you're not having antibiotics in the tea dip. And incidentally, while we were developing it as a post-milking tea dip for mastitis prevention, we found that it has great application for dry cow therapy, for which it has been marketed in Europe. So while we are addressing productivity, we are simultaneously addressing the safety of the products. So coming to the first, that is Filariasis, where we have developed, I will say, SLN or solid lipid nanoparticles. Now, the major challenge with Filariasis is it affects the genetically crossbred cattle and therefore is a major constraint in implementing the genetic improvement program, with a loss of about 3.8 billion rupees in India alone. Importantly, we have to understand that Filariasis is localized mainly in the liver, lymph node, spleen, kidney, lung. So if I can have a system that could go majorly to these organs, then I would have a targeted system. Now, the treatment currently for Filariasis is buparvacone as an intramuscular injection. But it is seen that this infection often recurs and becomes chronic, and the drug is secreted in milk for a few days, and hence the milk is unusable for that period. You need a withdrawal period, and a withdrawal period becomes expensive for a small dairy farmer. So we are going to look at targeting. So when I talk of targeting, what are we addressing? We are addressing two types of targeting that we can use. One is passive and the other is active. And as you can see here in passive targeting, that boat is put into a river and it just goes along with the waves. So in passive targeting, we put the system into the bloodstream and allow the body to take it wherever it wants. But for this, what is important is we have understood the body physiology, and therefore, even though I am putting my system into the bloodstream like a simple intravenous injection, I can predict where it will go, and I'll show you some data. In this talk, we are not addressing active targeting, which gives us better targeting. Because in terms of technology, passive is simpler, it's easier, and more affordable. So what is the principle of passive targeting? We are targeting to the reticuloendothelial system, which is a major physiological barrier. The major organs of the RES are the liver, spleen, lungs, bone marrow, and lymph node, etc. And here you can see what happens. You have a parasite taken into the RES or a macrophage. This is a live picture that you see here, and this is a cartoon. So basically, this is the way the body tackles bacteria, viruses, etc. So we use the same technology or the same principles of physiology for the body to take up the particles we deliver. And here is a picture of a particle actually being taken up by phagocytosis. So this is actin-based phagocytosis, where the actin film is rapidly forming. So this is something that is known. In literature, there is enough proof that phagocytosis can be relied on to deliver particulate systems, in particular nanosystems also, to the reticuloendothelial system. So now, why did we choose solid lipid nanoparticles? Because if you open literature, you will find a lot of talk about liposomes. But liposomes are made up of phospholipids that are very, very, very expensive. It is not even affordable for humans across all socioeconomic strata. So here we chose lipids which are safe, which are hydrophobic, because that is important to have RES uptake. Low cost lipids are chosen, and this technology is easily scalable. There are methods that are reported. So, but even though we say easily scalable, it is with reference to liposomes. So they are an attractive carrier system and affordable carrier system. So how are they made? Just to give you a gist. You melt the system, or More often, you can just dissolve the drug and a lipid because it's a solid lipid nanoparticle. You then add it to an aqueous dispersion. It is homogenized using an ultraterax. Then again, it has to be high pressure homogenized, finally freeze dried, and you get a powder. This powder can be reconstituted a normal saline, dextrose injection and injected to a human being, or now we're talking of buparvacone, so to the animal. So you can even reconstitute it in 500 ml of dextrose saline and do a slow injection. The particle size we got of an optimized system was roughly 580 nanometers. This says it is acceptable because PDI was less than 0.4. What is important, we got high entrapment efficiency. So about 80% of the drug went into our system and gave us a drug loading of about 40%. This is a picture that shows you, it's not one of the best pictures I have, but here we saw that all the particles were round. So we got solid lipid nanoparticles or SLN that were round. And now I will take you through some studies that we have done with it to show that it is targeted and that it works clinically. So this was studied first for safety. So we saw less than 8% hemolysis, which says it's a good system in terms of intravenous administration. It is safe for IV administration. We also needed to do, since it's a nano system and nano systems can agglomerate quickly within 10 minutes also, then they can block a blood vessel and cause instantaneous death. So to prevent this occlusion, we also checked the in vitro serum stability, and we saw in vitro serum stability up to 120 minutes. I'm showing you that we are following detailed procedures, but I'm not going to detail the procedures out to you so that I take you through the more exciting parts. Detailed procedures, if anybody is interested at any point of time, I would be willing to share one-to-one. Then it was important because we say it's taken up by the RES, so we needed to look at macrophage uptake. So we looked at the U937 cell line, and we had to make sure that our system is not cytotoxic and that the cells remain alive. So we found that when we looked at 1 hour, we found that both the drug and the SLN were not really cytotoxic, even up to concentrations of about 25 microgram per ml. And at 24 hours, of course, at 2 hours, it was very good. At 5 hours, 10 hours, etc. Up to 10 hours, the SLN was still good, but the BPQ was showing toxicity. So we say that BPQ, SLN are safe. And we could do these studies because you don't do intramacrophage uptake over 10 hours. You just do it over 1 hour. You can even stop it at 6 hours, etc. So here is some data that we saw. What we did is we took this SLN and we tagged it with coumarin, and we monitored under a fluorescence microscope, and we saw that with time, slowly, the fluorescence increased, which means particles are taken up. I think you can see these particles, and here you can see that they are quite round also. And we monitored it by quantifying the drug, and we found that at the end of 1 hour, about 52% of the drug we had given was taken up, which said it's really undergoing good macrophage uptake. And we could quantify the drug, and the drug had not degraded also. So it tells us that the drug would be available for action. And in terms of technology, the system was stable for a year, which is considered reasonably good. We tried for 2 years, but here, since we have to move ahead with the PhD project, once we got 1 year stability, we said we are good to go forward. Then we did the biodistribution. Now, I'm not showing you the basic biodistribution protocols of labeling and assessing stability of labeling. We've done all that. We followed all procedures because we had guidance from Dr. Samad and Dr. Gaikwad and also Dr. Galdhar for this. So here are scintigraphic images revealed high accumulation in the liver. So the scintigraphic image could not distinguish between liver, spleen, lungs, etc. But if you look at this graph, you will see that we have high accumulation in the lungs, in the liver, lungs, spleen. We also see some accumulation in the lymph node, and in the blood, the accumulation is low, which means most of it has gone into various organs. And this was by scintigraphy. So we also actually quantified the drug by HPLC method over a period of 24 hours. And these were the organs we looked at: liver, lungs, spleen, heart, kidney, and brain. And here you can see that liver and spleen, that's the blue and the green, show us very high uptake. Again, here liver and spleen are higher than all. So this showed us an RES targeting efficiency of greater than 85%, which means 85% of the drug we had administered had gone to the major RES organs. And the equation you see below is what was used to calculate this targeting efficiency. Now, what we did was next the efficacy study. Because very important, what we're looking at is a system that has to work in the clinical situation. So this study, again, like I said, the entire thing was initiated, supported. So it's a total collaborative study where we did the technology, and for all the other scintigraphy, clinical, etc., it was a responsibility of the Vet College, Mumbai Veterinary College, and all our collaborators, and we worked together. So we had exclusion criteria where animals treated with a drug earlier, up to 3 months, were excluded. We had inclusion criteria which said high fever, anorexia, and swelling of the superficial lymph node. And then PCR, lymph node biopsy, etc. were performed. And what we did is, since we were injecting a targeted formulation and the intramuscular route is 2.5 mg per kg, we went to a very low dose. So 0.25, that is 10%, and 0.5 mg per kg, that is 20%. And then we looked at the rectal temperature, hematology, lymph node biopsy, PCR, all that on 0, 6, and 30th day, and also lymph node size. Now, what were the signs we looked at? There are some pictures for emaciation, sticky discharge from the nose, paleness of the vaginal mucous membrane, enlargement of the prescapular lymph node. You can see that very clearly here, opacity of the eye, and blood in the stools, that is blood in the dung. These were clinical parameters. And like I said, a dose of 0.25 mg or 0.5 mg per body weight was dispersed in 5% dextrose 1 liter and administered at a rate of 5 ml per minute. So it took about 20 minutes, or no, it took quite some time. It was a very slow administration done by a competent veterinary doctor. This is a freeze-dried SLN that was dispersed. So we ensured that the dispersion would occur very easily, no agglomerates would remain, and the safety was already studied before we handed it over for the study. Now, measurement of lymph node was carried out, a lymph node biopsy was carried out, a blood smear was stained and evaluated for the parasite, and rectal temperature was also monitored over a period of time, in addition to various other parameters that you can see in this slide. So this slide shows you the 0, the 7, and 30th day for a 0.25 mg per kg of our SLN, 0.5 mg per kg of the SLN versus 2.5 mg per kg of Butalex. Now, if you look at this data, it will take you a lot of time, but I will summarize it for you that the data for all the three systems was comparable. So which said that even at 0.25, our SLN seems to be working well. So there was good clinical recovery also seen in the parameters that I showed you earlier, and there was no significant change in these parameters that monitor hepatotoxicity. And therefore, since our system goes a lot to the liver, this was very critical parameter, and no hepatotoxicity told us that it is safe for administration, because BPQ as Butalex IM may not go so much to the liver. So this was a very critical finding for us, suggesting that the system is good. But when we did the whole blood PCR test, we saw that at 0.25, they were all still positive, but at 0.5 and with Butalex, they were negative. So this told us that a practical dose would be 0.5 mg per kg and not 0.25 mg per kg. So we decided to select 0.5 mg per kg as our dose. We also monitored, like I told you, the lymph node size, and that was done at 0.5 mg per kg. So the left and the right were monitored. Now, if you look here, you find that this is at 0.5 mg per kg. This one is 0.5, this is 0.25, then this is 0.5, this is 0.25, this is 0.5, this is Butalex, 2.5, left and right. So this is all right, this is all left. So basically, we saw a significant reduction in the lymph node size with all. Probably at the 7th day, Butalex seemed to be a little better, but we also found that our system gives you a significant reduction, and by 1 month, the reduction was comparable, which means the system was working and actually helping to reduce the lymph node size over a period of time. Now, everything was not wonderful. We did see some adverse reaction at the 0.5 mg per kg body weight, but at 0.25, we saw the PCR was positive. So at 0.25, we saw reddening of teeth. We saw edematous swelling of the vagina and of the eye. However, these reactions were readily overcome by AVEL intramuscular injection. And therefore, and this was not seen in all the animals we tested. It was seen only in some, so it was still considered acceptable. So what we would like to say here is that our SLN at 0.5 mg per kg, that is 5 times lower doses, was as effective as Butalex. So this is something that we found very interesting, very exciting, and we have an Indian granted patent on this, which is already licensed to Sci-Fi VetMed Private Limited in New Delhi. What was our next step? Now we have this license, which means we have to commercialize, we have to support in the commercialization. So I'm just showing you the steps here involved if I have to commercialize it in terms of only the technology. Because we're showing it works, and this company was convinced that it works, and they were willing to take it forward for translation. So we dissolve the excipients in an organic solvent, precipitate it out as a nano system. We remove the solvent, and then if required, we have to do a size reduction. Then we have to isolate, then we have to lyophilize. Each of these steps in terms of technology is very, very involved, and from small scale to scale up are huge, huge challenges. So we got wondering, we started it and we have done a scale up to 100 grams, not of this system, but another system, because we are working on a number of systems. But then we said, if we have to go to kilos and a large batch size, how do we do it? And that's when we came up with this very wonderful idea, which we call as in situ buparvacone SLN, which is a very simple technology to address complex biology. So if you see here, you have so many hurdles that you have to keep jumping. And let me tell you, each one is a very difficult hurdle to cross in itself. So here we created a system that could just jump past and reach the goal. And how did we do this? We prepared a simple solution of lipid and drug with stabilizers. That is what is unique here. So we have only a solution which, when added to an aqueous medium, for instance, this is my injection, which is a solution. So all of us know how easy it is to prepare. And this was done by my student Hina, Dr. Hina Maithenia, who's now in the US. So you just take this injection out. You can see it's a solution. It is added into 5% dextrose injection. And can you see the formation of the nanoparticles? You just shake it like this, and then we have a ready to inject BPQ SLN. So while we started by doing it by methods that are reported in literature, this is a method we evolved in our laboratory. And very important for us to understand that with this method, what we have is a simple and green technology, no solvents, instantaneous formation of nanoparticles, minimal energy, because I don't need a freeze dryer or a filtration assembly or a homogenization assembly. I just need a stirrer and a tank to dissolve and then a sterilization filter or autoclave. I can process heat labile drugs by filtration, sterilization. All stability issues are bypassed totally. It's cost effective because my infrastructure is very simple. It's industrially scalable without a challenge. And what we got using this very simple method is a size of about 600, you see. We didn't do anything except dilution of a solution. That composition is very critical, and that's where our technology expertise comes in. Particle size was in the range that we're looking at. Drug loading was good, but look at this. If I have put 100 mg of drug in that system, in my solution, 97 was going into the particles and only 3 was outside. And even when you freeze dry and you disperse in dextrose, about up to 5% does come out into solution. So we are absolutely within the limits of what is prescribed by standard nanotechnology. This is a picture of the particles. Now what we noticed here, surprisingly, is they were not round. They were elongated, they were a little flattened, and they had rounder edges. So this, if you see this one particle, you'll see it's not round. If you see this particle, you'll see it's not round. This particle is certainly not round. So the particles were not round. And they had an aspect ratio of about 2.7. And we measured about 1,000 particles before we arrived at this value. Just to tell you, in this case, we went through a complete stability study and established a stability for up to 2 years, which is very good from the commercial launching perspective. Again, we did the in vitro serum stability because this was made by a totally different method. So we did the serum stability and we did not see aggregation for up to 6 hours because our system here became even more stable the way we had designed the technology. And hemolysis was low. It was just about 8 to 10%, like the standard nanotechnology. And the cutoff is 20%. So we are well below 20%, which says, again, we have a safe to inject system by intravenous route. So where are we with the biodistribution? Again, we did see extensive distribution in the RES tissues, but I'll show you some data. Now we looked at biodistribution of free technetium. And if you see free technetium, this is the thyroid, this is the stomach, and this is the bladder, and this is the intestine. So that is where you are seeing a lot of free technetium. But come to the BPQ SLN, we see a lot of it in the spleen, in the liver, in the kidney, in the lungs, in the lymph nodes also, over a period of 6 hours. Surprisingly, we saw that compared to liver, we are seeing very high concentration in the spleen, and the total RES accumulation even here was more than 75%, which is excellent. So here what we did is we looked at the spleen-liver ratio over a period of 6 hours, and we saw an astounding ratio of greater than 11 at 3 hours. So at 1 hour, it was about 9, then it went to 11, and even at 6 hours, it was more than 4, saying that this odd shape helped it to accumulate a lot in the spleen. And in fact, we were surprised to see these specially differently shaped particles, and that they give splenic accumulation. And the infections, incidentally, that we were looking at, Filariasis, brucellosis, etc., are highly spleen resident, and the biggest challenge is sterilizing the spleen. So through such a system, probably we can get excellent sterilization of the spleen and the liver because the drug is accumulating to a large extent compared to conventional in the spleen. So now let's move on to the clinical studies, which we did in collaboration with the Postgraduate Institute of Veterinary Sciences in Akola. And again, we had inclusion criteria, exclusion criteria defined by the college, but the dose we selected here now was only 0.5 mg per kg because we were permitted to do a very limited study. Again, some monitoring parameters were rectal temperature, hematology, lymph node size, serum biochemistry, before and after the treatment. So if you look at this This is before treatment and this is after treatment. So here, rectal temperature, because fever is one of the clinical symptoms, you can see it went down to 101. Heart rate went down to 92 and hemoglobin jumped up, which means the general health status is certainly going up. We did see a lot of other parameters, but we didn't see too many changes, saying it's not affecting too many other parameters. But in these parameters like total erythrocyte count and leukocyte count, we did see a significant difference. In the others, there was no significant difference. But in general, we found that clinical symptoms showed a marked improvement in the general body condition. The appetite and heart rate showed improvement, which is very important because appetite is generally an indicator of good health, not only for the animal, but for humans also. So we appreciate that also. Significant decrease in rectal temperature suggested recovery of the animal from the infection. An improvement in total erythrocyte and leukocyte count also is an indicator of recovery. Importantly, at the end of 3 months, with Butalex, recurrence has been observed, but in our case, we did not see recurrence in a single animal because all of them were monitored up to 3 months. Now, since we are talking of food safety, meaning product safety produced by the animal, we also did a milk residue test. And we did all this because we were looking at giving this also to the industry and telling them go ahead with this first, and then maybe we can optimize the other one. So the maximum residue that was seen in milk over a period of 48 hours was 6.15 nanogram per ml, while the FSI allows 10 nanogram per ml. So we were well below. This was a limited study in only 4 animals, but in all the 4, we were nowhere close to 10, suggesting that there may be no withdrawal period and milking can continue. So there is going to be no adverse effect for the farmer in times of losing his daily income, particularly small and marginal farmer. So what are the highlights of this particular study? This particular study tells us that we have BPQ SLN by a very simple and innovative technology to address something quite complex like Teniosis, which is really a challenge because it is seen to afflict over 25 to 30%, particularly of the genetically crossbred cattle. We see very good therapeutic potential at a significantly lower rate and no exuding in the milk. So that also tells us is a major advantage of targeted delivery of buparvacone through the SLN formulation. And a third important thing, particularly with the in situ SLN, is that it could be a promising splenotropic system with good translational potential and improved efficacy. So where are we today? We have a product that is licensed. This is the proposed product and package. It is called Buantrap. The technology is licensed again to Sci-Fi VetMed Private Limited. And incidentally, the managing director of Sci-Fi VetMed is also a veterinarian, Dr. Ali Asgar, and probably he's passed out from Mumbai Vet College. I'm not certain about where he passed out from. We have a patent filed on this and we are expecting it to be granted soon. So with this splenotropy that we saw and many intracellular critical infections like brucellosis also being splenotropy, I will just show you a little bit of study that we had done on designer nanocarriers for tailored splenotropic delivery, where we worked on doxycycline hydrochloride, and I call it lipomer because it is not just an SLN made of lipid, but it is a lipid and a polymer. So like I said, technology also is challenging because doxycycline hydrochloride does not get entrapped in the lipid because it's very water soluble. So then we came up with this lipid polymer combination, which is in itself something exciting, but I'm not showing you all the data we did to design the lipomer. But very important, I would just like to tell you a very small story about this, is that we started to design this doxycycline hydrochloride lipomer for RES targeting. And we suddenly found that with a particular system, we saw very extensive splenic targeting. So I'm going to show you just that part of the study. And this study initiated by Dr. Rajesh Patil, who's also in the US now. And he's the one who started the study on doxycycline lipomer, optimized it to get very good drug loading, good size. And here are some pictures. I'm showing you single particles so that you understand the shape. So we had 12.5% lipid, 25% lipid, 37.5% lipid. And as the lipid increased, the shape changed. And it became really asymmetric, irregular, if I may say so. And here you'll see we have a paper with Dr. Gaikwad, Dr. Samad, all of them as co-authors with us. So with 25% lipid, we could also make these round particles. So that's why we call them as tailoring the nanoparticle shape. Now, what are we looking at here? This is the scintigraphic data that we are seeing. This is organ-associated radioactivity in the liver, which is blue, and spleen. So this particular, we had a lot of systems where all of them showed more in the liver, less in the spleen, and a lot in RES targeting of greater than 75%, which is very common. But this particular system that we had with this lipid called glyceryl monostearate showed us very high splenotropic delivery compared to liver. So the spleen level was much higher. And if you compare this 12.5, this is that 12.5 lipid particle. It's comparable. But the minute I went to 25, this particular shape, I saw it going up like this. In the spleen, when I went to an even more irregular shape, the spleen concentration went further up. And this is the round shape, which is here. And I see the spleen concentration coming down, which tells us that shape is directing my particle to the spleen. Now, I'm telling it to you very simplistically, but to find out that it was shape took us almost 6 to 8 months. Because this is not reported anywhere. And we were the first to find this. And thanks to Rajesh, who realized that there is such high spleen concentration, that the study took a fantastic turn. So as we increased and the shape changed to more irregular, we got more and more splenic uptake. So now, this is the one particle here, which is not showing splenic uptake. So which means an irregular particle goes to the spleen more and a regular particle round goes more to the liver. Pardon? 10 minutes? Okay. Then we did a spleen. 10. So we had scintigraphic done in rabbits. Somebody is talking. Can that person mute, please? It's very distracting.
हेलो
सर भी बोलेंगे कि वो क्या बोलता है पूछ के बताओ तो अच्छा पहले पूछ लो
हेलो
Please mute.
हेलो राखी प्लीज म्यूट राखी भारती हाँ किया
Okay. So we did this spleen liver concentration. We wanted to check it out because the first studies were in rats. When we did it in mice, also we saw a slight increase in spleen, which was not too significant, but it was equal in liver and spleen almost. It was 1 point something. But in the rabbit, it was 2.86, the spleen-liver ratio. Then we did it in dogs, and here we actually monitored. So I'm going to show you some dynamic scintigraphic images. This is a spleen. Can you see it's going a lot to the spleen? And this is something we monitored in the first 5 minutes, and we're showing you a small video clip. Now, this was that irregular lipomer, and this is the round one at 25%. And this is a spleen, and you'll see there's hardly any dark radioactivity. So this is how it finally, this is a picture. So with the irregular lipomer, we got a ratio of 6.77, and with the round, we got 0.538. So this confirmed for us that if I have an irregular shape, I am going to get high spleen-liver ratio. This is just summarizing the data. And this study, part of the study on the shape was done by Dr. Anil Jindal, who is an assistant professor in Rajasthan in Pilani, BITS Pilani. So here also the paper is jointly with Dr. Samad, Dr. Gaikwad, etc. And since at that point of time, the brucellosis infected animals were not available, the study was done with Dr. Gaikwad and Dr. Samad in dogs suffering from ehrlichiosis, for which this drug also works. And some clinical parameters were monitored like platelet, WPC, spleen size, fever resolution, and regaining of appetite. And here is some data that shows you an increase in platelet count was seen, but no significant decrease was seen in antibody titer value. But WBC, there was no change seen. However, there was a significant reduction in the spleen size. Here you can see a huge swelling which became almost normal after 5 days of treatment with our doxycycline hydrochloride lipomer, which was at 10% the dose, 1 milligram per kg intravenous versus conventional oral 10 mg per kg, and an 83% efficacy with all the dogs recovering well. And in fact, we were told that a lot of pet owners started coming to them, asking them, but we have not been giving them the samples for ethical reasons. But it is a system that has worked beautifully. Now, to take these systems to translation, whether it is the SLN or the lipomer, we have done all these studies, and these were done in collaboration with Dr. Geeta Vanage, Deputy Director. Now she's retired at the NIRRH, that is the National Institute for Research in Reproductive Health. So we did the acute and subacute toxicity as per OECD guideline, genotoxicity, 3 tests, mutagenicity, and also we went ahead and did the developmental toxicity because we would be working on fertile animals and we want animals to produce milk, and therefore this was very critical. All these studies showed us that our system was safe in the concentration we were going to use. So I'm not showing you that data, it's vast data. So the maximum tolerable dose, this is for BPQ SLN, was 75 mg per kg. And the NOAEL was 15 mg per kg. So we are going to work at 0.5 mg per kg, which is very low. Acute toxicity, this is of the lipomer, was 6 mg per kg. So, and the NOAEL was also 6 mg, and we plan to work at 1 or 1.5 mg per kg, which is again lower than this. Here is some data on the developmental toxicity, where if you look at this talks of the fertility index and the pre-implantation loss. So what we see that there is no significant change in the fertility index and pre-implantation loss compared to the vehicle control, particularly with the blank SLN, which meant that the system we were using, the carrier material was very safe. But when you look at this, there is some loss, but it was considered that this is going to be better than the other system that is commercially available right now. But in general, no skeletal and cartilage deformity was seen in the fetus, which says that it would not affect the fetus. So this is some developmental toxicity with the BPQ SLN, and no teratogenicity was also observed with both the BPQ SLN and the blank SLN, as all these are very, very critical considering the animal group we are discussing. And then BPQ SLN and blank revealed no genotoxicity, no skeletal abnormality, very limited effect on fertility, and non-mutagenic. They were also non-mutagenic when studied by the AIDS test. This is for the lipomer. Similarly, we find no skeletal deformity. And we find here with this that there's hardly any change in both fertility index and post-implantation loss, suggesting that the system is very good. So here again, our conclusions are same. Now, here what we found that when we went to very high doses of 15 mg per kg, etc., we did see some challenge here. But up to 6 mg per kg, we did not see any challenge. And no genotoxicity was seen at 1.5 mg, which is a dose that we are planning to use. No skeletal abnormality and no change in pre-implantation loss, fertility index, etc., suggesting that we have come up with good systems, practical systems, and commercializable systems. And like I said, the buparvacone SLN, bu-ant-trap, would have been commercialized if it were not for the COVID pandemic. But I just had a discussion with the collaborator last week, And I'm told that by 2023, mid-2023, that product is likely to be launched. And now I'll come to another very innovative formulation, just 2 or 3 slides. I won't take more than 5 minutes for this, which was designed for mastitis, which is a very costly problem. And if we understand correctly, mastitis is one of the biggest challenges for the dairy industry, because it significantly affects milk production. Up to 70% loss in milk production is possible when animals are infected with mastitis. And unfortunately, there is no simple solution, so prevention is the goal. So looking at prevention, what we worked on was a post-milking teat dip, which we found that also works at drying off. And I'm only going to show you a picture of the teat dip here. Now this is a teat dip. Can you see this plug? This plug forms reproducibly on all the teats. And this plug forms simply by dipping. You don't need to do anything special. We dip it and then we remove the cup, and a plug is formed on every teeth, and that efficiency is 99%, or I may even say 100%, which forms spontaneously. Now what happens is this plug is very strong and hard. It also protects the teeth canal, gives additional teeth protection, and therefore we realized it could also work for dry cow therapy, where currently you have injectable implants which are injected into each orifice. This is so simple, you just dip. So this innovation, developed as a post-milking teeth dip, became something applicable for a very important treatment, not just prevention of mastitis in between milking, but prevention of mastitis during drying off. And it can remain there for 3 to 5 days without a challenge, even if the animal sits on the floor, gets up, etc. At the same time, it's readily washed off when you want to remove it. So that is a very important advantage. This is the product that is launched in UK and the European market. So it's called Ubera Dry. Very attractive package has been made by the industry partner. We have an Indian patent already granted for this, titled Bioadhesive Barrier Film and Spontaneous Plug Forming Teeth Dips. So it's called a barrier film, and I would like to point out it does not have any drug. So there is no antibiotic or no drug in this, which is again something that is very important, because teeth dips that have antibiotics give you a challenge of milk contamination with the antibiotic. And again, the balanced teeth dip that falls, etc., is going to take that antibiotic into the drains. The patent is licensed, like I said, to Sci-Fi VetMed. This product is launched for dry cow therapy, prepartum intramammary infections, and summer mastitis. I have heard it's doing very well. So in UK and in Europe, it's launched in France, Belgium, Spain, and Finland. And probably the next point where it would be launched is New Zealand, that is outside Europe. So it's going very strong. So what I would like to say is, innovative formulations present a practical and very attractive strategy to enable enhanced health and productivity of livestock, as well as they would address safety of the food products for human use. So I would like to thank my collaborators, Dr. Abdul Samad, retired Dean and Director Mafsu. I saw his picture when they were all moving earlier in the initial part, Dr. Gaikwad, Dr. Geeta Vange, and my collaboration with Dr. Samad continues even today. Also with Dr. Gaikwad, we have been talking a lot and applying for joint funds together. This is the institute I come from, the Institute of Chemical Technology, which will soon be 90 years old, probably as old as Mumbai Vet College. And importantly, we have now 2 more campuses, one in Bhubaneswar outside Maharashtra and one in Jalna in Maharashtra. And these campuses were started only in 2018, so they are just about 4 years old. This is my research group, which is very important for me. Here is only a group that was here at this point of time, but I owe huge thanks to all my students over the years, because the work I presented is not really current work. Part of it is current and part of it is what has happened earlier. So huge thanks to all my students for their enthusiasm and the vibrancy they bring into the lab and the excitement that they bring into the lab. So thank you very much for this opportunity. Thank you.
Thank you very much, madam, for going through, taking us through a very systematic that journey of innovative whatever formulations are there. I really we appreciate your great contribution in the field of this innovation, and that too for a veterinary field. So it's very appreciating. So now I just request the participants if they have any question, 2-3 questions they can ask, please.
I'd be happy to answer questions. So I know people have listened to me.
Yeah, madam, very much. But you explained in such a simple way and very, very simple and very understandable that manner. Therefore, I think they have understood a lot of things. But our one of our staff working in the project, he has one question.
Yeah.
That whatever that spontaneous plug forming drug barrier, whatever you told. So that plug is used for how many days? He is asking.
It stays for 3 to 5 days.
Huh.
And in that time period, the teeth canal automatically closes during drying off. So it stays for the duration required.
Okay. Okay.
That's how it's gone commercial, no? Because that has been shown. The company has checked for itself that it works, and then they have licensed it out from it's licensed by Sci-Fi Vetmed to another company, Orion, and that company has checked everything and then launched it in the market. No, it's available. If you go to the website, you will see Ubera Dry being sold online today.
Okay. So these whatever developments are there, they are very, very important for us because all these whatever Theileria, brucellosis and mastitis, those are the major problem which we are facing, madam. And really we have contributed a lot in that, and hope again in future also we will have some more again upgradation and more that better technology again to improve. So I once again thank you for accepting our request and delivering a very nice speech today. So we thank you very much, madam. Thank you.
Thank you so much.
Thank you.
Thank you, madam, for very informative and excellent presentation information. Because as we have seen all these slides, which is whatever you have mentioned in that, which is related to veterinary feed, and that is very important because that is recent information. Whatever you have presented, that will be very much useful to us. Thanks a lot, madam. Yeah.
So
thank you.
More than I think in between there were more than 175 participants. They got benefited with your speech, madam. Thank you so much for accepting and sparing your time, valuable time. Thank you so much.
Thank you. Thank you.
Now today we have second speaker, Dr. P.S. Banerjee sir, and he is working as station in charge, Eastern Region Station ICR IVRI, Kolkata, West Bengal, and sir is going to present his talk on mid-born parasitic zoonosis. So before I request to sir for his presentation, I will give brief introduction about Dr. P.S. Banerjee. So Dr. Parth Sati Banerjee graduated with a BVS and H degree from Bidhan Chandra Krishi Vishwavidyalaya, West Bengal in 1985 and subsequently completed master's degree in veterinary parasitology from JNKVV Jabalpur in 1988. After serving as veterinary surgeon for 3 years in West Bengal, he joined as assistant professor in the Department of Veterinary Parasitology, GB Pant, University of Agriculture and Technology, Pantnagar in 1991 and was selected as associate professor in the year 1998. In the meantime, he completed PhD from GPPU A&T Pantnagar. He was selected as senior scientist in the division of parasitology, ICAR IVR Ijatnagar in the year 2005. Dr. Banerjee was selected as the head parasitology division in 2011. He has 31 years experience in teaching and research. His research interest includes molecular epidemiology and diagnosis of important parasitic diseases of domestic and wild animals. During his time at IVRI, he carried out research contributing to molecular epidemiology and diagnosis of Cryptosporidium parvum, Ehrlichia canis, Babesia gibsoni, Poultry coccidia, and Trichinella species. He was also actively involved in several disease investigations in wild animals and human trypanosomiasis. He has guided 10 MUSC and 4 PhD students and published more than 155 research articles in national and international journals. Apart from contributing in 7 books, chapters, 5 laboratory manuals, 1 mobile app both in English and Hindi, and 1 color atlas on parasites of veterinary importance. In recognition of his teaching excellence, he was awarded Best Teacher Award at GBPU A.T. Pantnagar in the year 2001 and 2, and same at IVRI Deemed University in 2014-15. He has handled several research projects, including one international project funded by BPSRC UK. He served as the editor-in-chief of Journal of Veterinary Parasitology, the official journal published by the Indian Association for the Advancement of Veterinary Parasitology during 2011 to 2015. Presently, he is serving as station in charge of Eastern Region Station, ICR IVRI, Kolkata. With this vast experience, I invite and request Sir to please deliver his talk. Sir, please.
Thank you very much, Dr. Vaidya. Now, will you allow me to share my PPT?
Yes, sir. Yes.
So now I am sharing. Is it visible?
Yes, sir. It's visible.
Whether my PPT is visible?
Yes, it's visible.
Now, may I go to the PPT mode?
Yes.
Okay.
Yeah.
Thank you very much, Dr. Vaidya, for very exaggerated, I should say, introduction. And I am a parasitologist, having the experience of working in the field as a veterinary surgeon for 3 years, then in teaching veterinary parasitology at GB Pant University, as you have seen, and then a little bit of research at IVRI Ijatnagar. So I have got experience of field as teacher and also a researcher. The topic allotted to me in this training program is mid-born parasitic zoonosis. I don't have any idea about the composition of the trainees, but I believe that it includes students, postgraduate students, and also faculty from different colleges of India. So with this, I like to introduce this zoonotic disease. The mid-born parasitic zoonosis today and the mid-born parasitic zoonosis 20 years back have changed a lot. There is a sea change, you can say, because of globalization, world trade. Now, if you wish, you can buy a Chinese preparation imported or a Japanese preparation in the restaurant or in through online mode. And this actually paved the way for mid-born parasitic zoonosis in a very diverse way. The parasites which are not present in India, those which do not propagate in India, but still we can acquire the infection from imported meat. simultaneously, you have seen migration of animals from across the continent. A few days back, 8 cheetahs have been introduced to India. We are not sure about the whole parasitic fauna. It is not possible to assess the animals, particularly the wild animals introduced to our country. And there is a chance of introduction of new parasites in the wild. So we should be aware about all these parasitic infections which may spill over to the humans through meat. Oh, what are the factors that are responsible? As I was telling, that globalization has paved the way for introduction of many parasites in our country. And if we look at other factors like ethnic food habit, the people who are migrating to India from other countries, we have seen in the past that there was a mass migration from Vietnam. There is also a mass migration from some other East Asian countries to India, and they have got their own food habits. And food habits hardly die, as bad habits die very hard. So the food habits cannot be changed, and it is very difficult to change the food habit of a ethnic group. So this type of food habits actually is responsible mostly for the propagation of meat-borne parasitic infection. Then, which includes improper cooking. Cooking kills the flavor. This is the perception in a particular group of people, and there is method of preparation of meat which is not sufficient to kill all the contaminants, including parasites in it. So improper cooking is a major factor. Sanitation of the human population, the Swachh Bharat mission actually stressed to the prevention of or vanishing the defecation in the open field, and it will greatly reduce many of the zoonotic diseases. So this was a superb step taken by the government of India, and in coming 10 years, it will yield the results. Animal rearing system is another very important factor. We know that there are many animal rearing systems, including intensive management under controlled condition, or a free range system where the animals roam about in the open spaces and there is no boundary. They can roam here and there, and this type of system will encourage many of the parasitic zoonosis. Immigration, tourists, and illegal importation. I have already mentioned about immigration. There are tourists all over the country from different European and Asian and African continents, and their migration in these or their presence in our country may introduce new niche of parasitic zoonosis. Illegal importation is another very important factor, and the people have developed rearing some exotic birds or animals, and most of the time they are illegally imported, and this is another risk factor. Lack of awareness is the most important one, and it has been shown that when awareness was created amongst rural people in Mexico, the cases of Neurocystic Sarcosis and intestinal teniosis decreased greatly. So awareness can cause havoc, and it can influence the prevalence of the infection. Then there are many economic factors which are related to social structure, and this is also very important. Lack of, lastly but not the least, lack of political will and commitment. This is the another very important issue. If there are laws, then there must be some will to implement it, and many a times we are lagging behind in this front. So these are the factors responsible, and we are almost sometimes buying the zoonosis at the cost of our life or man force. Now, while doing preparation for this lecture, I was thinking that whether I should concentrate on the word meat loosely, talking about the meat we take, like the mutton, the shaven, the pork, the beef, etc., or I must also include the fish, crabs, shrimps, etc. So I just searched what is the definition of the meat, and meat is defined as the skeletal muscle and its associated tissues derived from the mammalian, avian, reptilian, amphibian, and aquatic species commonly harvested for human consumption. Edible offal consisting of the organs and non-skeletal muscle tissues also are considered as meat. This is the definition as per American Meat Science Association. So I have included in my lecture the meat-borne parasitic zoonosis, uh, all the organisms or the parasites which are transmitted through fish or marine animals like walrus, seal, whales, etc. These are delicacies and very costly affair, but people have got their own choice to go for it. Now, meat-borne parasitic zoonosis, let us see what are those infections which are common, which are important, and I have enlisted them as per the parasite and the animals which are involved and the source of infection. In this list, I have given priority to toxoplasmosis, and the parasite, all know it is Toxoplasma gondii. And the animals which are involved are the all domestic animals, especially the cats and other felines. So this parasite is peculiar because it has got a wide, very wide intermediate host range like cattle, buffalo, sheep, goat, pig, and many other birds even. And the source of infection for humans is the food and water contaminated with the oocysts shed by the cats and other felines. And this is a major source of infection for those who adopt gardening or come in contact with the soil, or those who favor the green vegetables to be taken as salad. And there is another mode of infection, source of infection rather, that is the undercooked meat containing the tissue cysts. I will come to it a bit later, and like to clear the air about how the infection takes place in humans. Additionally, organ transplantation, if the organ has got a tissue cysts of toxoplasma, then in the transplanted patient, who generally is kept under immunosuppression by medication, there the bradyzoids may come out of the cysts of the rupture and can cause acute toxoplasmosis. Blood transfusion is also another way, because if there are tachyzoids multiplying in the cells, particularly the macrophages, then they may set up infection in after blood transfusion in the recipient. Another very important way is the infected mother to the fetus. This has got serious consequences. I will touch a little bit about how it is transmitted and what is the effect of congenital toxoplasmosis. Then comes another protozoa, sarcocystosis, though it is not that much serious, but cases have been reported And the species of parasite which are known to cause human infection from animals are Sarcocystis hominis and Sarcocystis hedorni. These two are from cattle meat, beef, and Sarcocystis sui hominis. This is from pork, and the pig is the host. Undercooked beef and pork containing tissue cysts are the source of infection. And I have seen when I was working at Pantnagar, I had the knack of collecting parasite for demonstration to the students, and often used to go to the slaughterhouses, and also for collection of parasites from the pigs. I used to go to the people who used to slaughter pigs in their backyard, and the rearing system was also free range system. I have seen the very young children, they they do some petty job in the slaughter process, and they just sharpening the knife or bringing a bucket full of water for washing the carcass. And often I have seen that they are rewarded with the half burnt carcass, because when in the rural areas the pig is slaughtered, after slaughtering the bristles are taken off with hand picking because they are sold, and after that they put the straw around the carcass and fire it. This fire actually uh, burn the smaller bristles. After that, they scratch the whole body with a knife. And at that time, I have seen the tail of the pig and also the ears, they are given to these children who take it raw with salt. And one such situation, I just was curious about the parasitic fauna in these children, and I collected stool sample from 20 such children who were involved in this type of practice. And to my surprise, I found that 14 of them were harboring either Tinea Solium or Sarcocystis oocysts. And in fecal examination, I could find these infections. So Sarcocystis hominis and Sarcocystis sui hominis, if the sarcocysts in muscles are ingested with undercooked beef or pork, then they undergo multiplication in the intestine of the humans and may cause little bit of diarrhea, vomition, and abdominal pain. Next in my list is teniosis and cystic. This is very, very important and very common in our country. And Tinea Solium, Tinea saginata, and now another species which have been incriminated as zoonosis is Tinea asiatica. This is restricted to some parts of the world only, not in other parts. And from India, there is no report of Tinea asiatica, though it has been reported from Nepal. So if it has reached Nepal from the East Asian countries, then there is every possibility of entering of this parasite to India too. The pig, cattle, and buffalo, they are the host animals which are involved, and the source of infection is either measly pork or measly beef. Another condition is neurocystic sarcosis, and for that, one don't have to be a non-vegetarian, because the eggs which are attached to the vegetables contaminated with the eggs of Tinea Solium may be ingested, and they may suffer from neurocystic sarcosis. So a vegetarian, totally purely vegetarian, may get the infection of cystic sarcosis by consumption of raw vegetables. Then another tapeworm is Diphyllobothrium latum, which is actually a fish-borne infection. and different carnivores act as the alternative host in the nature. Humans may get the infection by consumption of fish, either partially cooked or raw fish with their food. Continuing to that list, we have Paragonimosis, and this has been two species have been incriminated, and this infection has been reported from few times from Manipur and also Arunachal Pradesh, and where the wild carnivores, crabs and crayfish are involved, and the source of infection for humans are the crabs and crayfish containing the metacercary of Paragonimus. This is the lung fluke, and the infection of humans may be confused with tuberculosis, where in northeastern region tuberculosis is also very much prevalent. So in the differential diagnosis, Paragonimosis must be differentiated from tuberculosis for an effective treatment. So this has been reported, and the wild crabs and crayfish generally caught by the children or young adults are fired in the forest, and then they are taken. So this is a major source of infection. Wild carnivores also suffer, and with the defecation, the eggs come out and the snails act as intermediate host. So Paragonimus or lung fluke infection has already been established or in the northeastern states. Then a group of flukes like Clonorchis, Opistharchis, Metagonimus, Heterophyes, and Haplorchis, all these are trematodes, flukes. And some are, or some have been, a few of them have been reported from India too, like Heterophyes and Haplorchis. They are all fish-borne infections, and fish containing the metacercary of the parasite is the source of infection. Now, all of us attending this training lecture are aware that Clonorchis and Opistharchis, which are known as oriental liver fluke of humans, they are responsible for cholangiocarcinoma. This is a very peculiar thing, that the parasite is responsible for carcinoma, and it has been demonstrated in many East Asian countries, including Korea, Japan, and China. Another nematode which is gaining importance as zoonosis is Anisakis, and the disease condition is known as anisakiosis. The two genera, Anisakis and Pseudoterranova, these are the two genera which are actually parasites of marine animals, marine mammals like walrus, seal, and whales, and also some piscivorous birds. And these infections or the parasites in the intestine of these marine mammals, they excrete the eggs which come out with the feces of these mammals, and they are in the ocean water. And the larvae which hatch out from these eggs are taken up by the mollusks and fish, where they remain in the intestine. And when the fish is caught or the mollusks are caught for human consumption and they die, then the larvae migrate to the muscles. And people generally get the infection by eating undercooked or raw fish and cephalopod mollusks. Then one very important parasite, Trichinella, and the disease is known as Trichinellosis or Trichinosis in humans and animals. There are many species, at least 9 species and 3 genotypes. That's why I have not mentioned any particular species. And the animals which are involved, again, is a huge number of wild animals and domestic pigs and rats. And they actually maintain the cycle in the wild, and there is every possibility of intrusion of these parasites into domestic cycle. Source of infection, mostly it is pork containing the encysted larvae, but it has been shown that horse meat, bear meat, and also crocodile meat, which is gaining popularity in many of the African countries, may be a source of infection for humans. So I was involved in a bit of Trichinella research while staying at IVRI Ajatnagar along with my colleagues, Dr. Hira Ram and Dr. Rajat Garg, and they are continuing that research till now. And I have left IVRI Ajatnagar for Kolkata, but I keep track of the research which is going on. So, important parasitic zoonosis transmitted through meat, either meat of animals or meat of fish and sometimes birds are enlisted. Though it may not be exhaustive, there may be some other infections also, but of less importance. Now, I like to talk about a little bit of toxoplasmosis, and the source of infection, as I have told, is the ingestion of the oocyst. Actually, in the cycle, if you look at the cycle, then you will find that the tissue cysts, which are formed in the rats, mice, and also some birds, they may be ingested by a cat or other felids, and they have got a peculiar tendency to prey on the prey animals. Inside the intestine of the cat, it multiplies sexually and then asexually, and ultimately oocysts are formed, which are excreted in the feces. These oocysts are ingested by these animals, or it can also be ingested by domestic animals, or through green vegetables and salads, we may get the infection, and the humans thus get infected with the oocysts. Ingestion of tissue cysts. Yes, the animals which ingest these oocysts, their tissue cysts are formed in these domestic animals. And when we eat raw or undercooked meat of these animals, then also humans get infected. And with the salad, I have also remained. Contact with the cat feces is another source of infection through ingestion of the oocysts. Mother to fetus. If a pregnant lady gets a toxoplasma infection, depending upon the stage of pregnancy, the parasite may be transferred to the fetus, and the outcome of that transfer depends upon solely on the stage of the pregnancy. And in worst situation, there may be abortion, there may be hydrocephalus, a mental retardation, etc. in the newborn. Now, blood transfusion I have already mentioned, and also the organ transplantation through which they may be transferred to humans. It has also been shown that the toxoplasma, while multiplying in the humans, may also be transferred through the semen. So venereal transmission has also been proved in case of toxoplasmosis. And the mother to fetus transmission is known as vertical transmission. Now, toxoplasmosis outcome, a little bit I have mentioned that if it is an immunocompetent individual, a normal individual, then there may be transient flu-like symptom and uneventful recovery. But if the patient is immunocompromised, who has picked up the infection by any of the ways I mentioned, then there may be severe form of the disease and even fatality. Then congenital infection may lead to abortion or hydrocephalus or mental retardation and blindness. The economic impact of hydrocephalus, mental retardation, and blindness has been calculated in some of the European countries, because the care of this type of patients is very high. The care cost of these type of patients are very high. This is one photograph just to show you that hydrocephalus or mental retardation in the babies who are born alive, and that type of baby is mentally retarded throughout their life, and it has got tremendous impact on the livelihood of the parents as well as the cost involved. The parasite either in the muscle look like a tissue cyst. This is I have borrowed from one of the parasitologists of southern India, Dr. Sri Kumar. He did his PhD on toxoplasma and presented this slide to me. And this is the tissue cyst containing the bradyzoids, and the cyst wall is very, very thin. And this we inoculated the mice in our laboratory at Nagar, and in the macrophage, the parasites are multiplying up as a tachyzoid, and after some time they will rupture this macrophage and will come out and will infect another macrophage. This is the acute phase of the infection, and immunocompromised individuals in case of immunocompromised individuals, this multiplication goes on very fast, and almost all the macrophages will then be infected with toxoplasma. And you know, macrophages take part in immunity of our body. So the immune system will be further compromised. What are the risk factors in toxoplasma? Cat as a pet, especially semi-pet cats. Semi-pet, by the word I want to mean that the cats which remain in the home or the owner for some time and then goes out of the home, and they do whatever they wish, including hunting on the rats and then again coming back to the home. And this type of semi-pet cats are really very dangerous. If your pet cat is totally restricted to your house, then they hardly ever get any chance to pick up the infection. But the semi-pet cats, they are very dangerous. They can pick up the infection from outside and can transmit the oocysts to humans. Undercooked meat, pork, mutton, shaven, and chicken, they are again a risk factor. And there is a very common practice in the Indian household that the ladies who prepare the meat often test it before proper cooking for the salt concentration and the flavor. And that is a very risky proposition in case of the housewives. Raw vegetables, carrot, radish, tomato, cabbage, and onion, all come in contact with soil. And the cats, they have got a peculiar habit. I have not seen it in any other animal, but in cat, you will find that they generally find out a soft soil and then dig a hole and then dig some soil and defecate and then put some soil over it and cover the feces. So this habit actually renders the oocyst viable for a very, very long time. If the fecal sample or the feces is open to the sunlight, then there is a chance that the oocysts will be killed after some time. But if they are covered with soil, then their longevity is very long, very high. And this type of oocysts remain viable. And any of these vegetables which are cultivated in the soil, contaminated with the oocysts, get contaminated. And many a times I have seen people taking a tomato or a radish or a carrot without proper washing, just a cursory rubbing in the hand or the any cloth, and then they take it. And this is a dangerous activity which may lead them to toxoplasmosis. Activity involving handling of soil with bare hands. Gardening is a hobby which is often seen in many people, And this is again a very dangerous situation, particularly for the pregnant ladies. They should avoid gardening with their bare hands. Gloves should be worn. Kitchen utensils, if the meat is cut with a knife and that same knife is used for cutting vegetables or fruits, then the tissue cysts containing the bradykardioids may contaminate these fruits and vegetables, and they may be transferred to humans. Organ, blood donated by carrier donors, if they are infected and containing the tissue cysts, then they can transfer the infection to the recipients. Pregnant women and immunocompromised individuals are most vulnerable to this infection. Now I am coming to teniosis and cystic sarcosis. This is one of the most important meat-borne zoonosis in our country. And few years back, we were knowing about only two species, Tinea Solium and Tinea saginata. And this list, another parasite Tinea species has been added, and that is Tinea asiatica. I will show you some of the differentiating features of Tinea asiatica from Tinea Solium and Tinea saginata in subsequent slides. This is also a zoonotic parasite. Out of these, only Tinea Solium is most dangerous because we know that human may act as the intermediate host also for Tinea Solium. And if humans ingest the eggs of Tinea Solium through raw vegetables, then the eggs, after hatching in the intestine, may be circulated to different organs, including brain, where they form cystic sarcus, and it leads to clinical condition in humans, particularly epilepsy. And one of our sports person from the country suffered from neurocystic sarcosis, and he was treated successfully. Auto-infection has been proved in Tinea Solium infection also. Auto-infection means if there are adult tapeworms in the intestine, we know that the gravid proglottids are released from or detached from the rest of the strobila and are passed in the stool. And during this process, there may be a rupture of the segment in the intestine itself, and that will lead to infection of the same individual with the eggs of Tinea Solium, leading to cystic sarcosis. So this is a parasite where we find that humans may act as definitive host. At the same time, human may also act as intermediate host of this parasite. Now, I will show you that how Tinea Solium look like. It has got 4 suckers and also a well-developed rostellum with 2 rows of hooks. This is the scolex of Tinea Solium. And this is the heart of one pig, where you will find that the cystic sarcus are present in the heart muscles. So plenty of cystic sarcus in this heart muscle. And if this type of meat is ingested by humans before proper cooking, then adult tapeworms are formed in the intestine. This is a typical and very serious meat-borne zoonosis in our country. Here, a little bit of morphological differentiation of the 3 species. Tinea asiatica resemble Tinea Solium in some morphological front, and also Tinea saginata in some morphological front. Tinea Solium and Tinea asiatica, they utilizes pigs as their intermediate host, and Tinea saginata, it utilizes cattle and also buffaloes and some other wild ruminant as their intermediate host. On the basis of the morphology of the mature segment and gravid segment and scolex, we can differentiate these 3 species. It has got, Tinea Solium has got a well-developed rostellum with 2 rows of hooks. Tinea saginata has got 4 suckers but no rostrum nor hooks. Tinea asiatica has got 4 rostrum and a rudimentary, 4 suckers and a rudimentary rostrum with traces of hooks. As far as the gravid segment is concerned, on the basis of the lateral branches of uterus, we can differentiate. Differentiation of the species in a geographical area is important for implementation of control measure. And we know that Tinea Solium may cause Neurocystic Sarcosis, but these two tinea, Saginata and Asiatica, never causes Neurocystic Sarcosis. Tinea Asiatica forms cystic circus only in the liver of pig, while in case of Tinea Solium, cystic circus is formed in different muscles, skeletal muscles, and also cardiac muscles. So these are some of the differentiating features of the 3 species of tinea. Here are the photographs. This is Tinea Solium with a very well developed rostrum and 2 rows of hooks. This is Tinea saginata, the 4 suckers only, no rostrum, no hooks. And this is Tinea asiatica, 4 suckers and also trace of hooklets, not exactly hooks, rudimentary hooklets and a rudimentary rostrum. So on the basis of this morphology, they can be differentiated. As far as distribution of Asiatica is concerned, Tinea Solium and Tinea saginata are present in all the continent and the countries where pork and beef are eaten, and Tinea Asiatica has been reported only from these red spots, the Japan, South Korea, China, Taiwan, Thailand, and also Indonesia, and also Nepal. So there is every possibility of entry into our country. Now, how to make the meat safe if all these tapeworms, particularly tinea, are infecting the animals and people are ingesting, then how the meat can be safe? Proper cooking or frozen meat. If it is frozen also, then also the parasites can be killed. But in our country, that type of frozen meat is not always common, or the freezing of the meat is not practiced regularly. But if they can be frozen at minus 20 degrees centigrade or below, internal as well as external, for 7 days, or minus 35 degrees centigrade or below for 15 hours, then these parasites are killed. And if the beef, pork, or fish and other meat is cooked thoroughly and completely at a temperature of at least 63 degrees centigrade, completely means it is thoroughly cooked at least for 10 to 15 minutes, then also the parasites will be killed. Here, a note of caution is that the killing of the parasite is dependent upon the meat piece. The external temperature of the meat piece and the core temperature of the meat should be 63 degrees centigrade. Otherwise, any parasite which is at the center or at the core of that meat piece will not be killed. So this is very, very important. So the meat pieces, if they are small, then it is easier to kill the parasites which are transmitted through the meat. If it is very what I can say, large in size, then the outer surface temperature may reach 60 degree, 63 degree, but the inner core temperature may not reach 63 degree. So this is to be kept in mind for the meat science people who advocate the freezing or cooking the meat for making it safe. Now, some of the methods which are used in the livestock products technology, like curing, drying, smoking, or even microwaving the meat, it does not consistently kill the infective parasites. So even if they are cured or smoked, they are not safe. That is the point to be noted. Now I will come to one of the areas which is what I can say, this is not a very important aspect as far as meat-borne zoonosis in India is concerned, but the research were conducted at Mumbai Veterinary College in Dr. Vaidya's lab and also in Dr. Hiraram's lab at IVRI. They unraveled many unknown things about trichinellosis. There are sporadic reports from a variety of animals and also humans from the country. Here in this map, we just prepared this map, and you will look into West Bengal. It was in cat and civet cat. Similarly, from Maharashtra, the cat, the civet cat, and also field rat and bandicoot, all these animals were found infected sporadically, and twice in the domestic pigs they have recorded Trichinella infection, and it is in the 1970s. After that, in Dr. Vaidya's lab also, they found Trichinella infection in domestic pigs. Apart from that, human infection has been recorded from Karnataka, from Uttarakhand, from Punjab, particularly Chandigarh, and also Uttarakhand in different places. Most of them are from very remote villages of Uttarakhand. And it all came to light in 2010 when a small outbreak took place in Uttarakhand, in which 54 people in a village, they hunted one wild boar, and it was celebrated in a feast in that village, and all the 54 people, they consumed the pork, and depending upon the quantity of pork they got or the share of their pork, they were ill, they fell ill within few days of ingestion of the pork, and some of their condition was very serious, and they were hospitalized in different hospitals of Uttarakhand as well as Delhi. And there, around 10 people, 10 to 11 people, they died. The infection was so severe with typical clinical signs of swelling of the face, high eosinophilia, fever, body ache, etc. After that, after this event in 2010, the interest to Trichinella infection or Trichinella research was renewed, and at IVRI, the then director also instructed that the veterinarians in parasitology division should take up research on Trichinellosis. And my colleague Dr. Hiraram took up a institute project. I was also involved, and we were searching for Trichinella in pigs, but it was not very successful as we were looking in domestic pigs, taking the clue from Maharashtra's findings. We were searching pigs by different methods, particularly the tissue press technique, muscle press technique, and also muscle digestion technique. But we were unsuccessful for around 2 years. And this was not taken very seriously and very pleasantly by the administration, and we were pressed to go for the survey of the parasite in those villages where human cases were reported. Ultimately, one day, uh, two carcasses of wild boar, which were poached by the poachers, they were brought to IVRI for uh, postmortem and also the confirmation, and we examined the muscle pieces of these two wild boars, and one was positive. So this was the first observation of Trichinella by our team in IVRI. After that, we had the opportunity to examine the carcass of the dead animals of leopard, tiger, lion, and also foxes and hyena and crocodile at IVRI, being a national referral center for zoo animals and wild animals. We were fortunate enough to get these animals, and we found that 75% of the leopards and around 50% of the tigers, they are positive for Trichinella. And then after finding them, the next target was to find out that which species are they. And from India, whenever Trichinella has been identified, it was named as Trichinella spiralis without bothering about the techniques, molecular techniques, because morphologically it is not possible to differentiate the different species of Trichinella. And we also tried with electron microscopy also, but we could not differentiate the different species of Trichinella. But yes, on electron microscopy, the larvae which were destined for male or female category would be differentiated. So genetic characterization part of, okay, I am coming to it late. This is just the cycle which is going on. In a sylvatic cycle, the wild boar and also the carnivores, they pick up the infection from the wild boars or sometimes the wild rats. And when a carcass is there in the wild, even if it has decomposed thoroughly, totally, then also the Trichinella larvae remain alive in the muscle. This is not very usual thing, because as a parasitologist we know that when there is a death of the host, the parasite also dies. But in this case it is not true. The total decomposition of the carcass cannot even assure the death of the Trichinella. Now, these wild rats, they may enter into domestic cycle, because in the fringe areas where there is a interface of the domestic area and the forest area, the rats migrate to even domestic cycle, and they can introduce the infection in the domestic cycle. Once it is in the domestic cycle, then man may get the infection from the meat, and they can harbor the parasite and can suffer from the infection clinically, though man is the dead end host of this parasite. Then we found the encysted larvae in the muscles of different wild animals, and we also tried to recover the live parasites from the muscle by muscle digestion. And after digestion in acid pepsin of the muscle, we could recover a huge number of Trichinella larvae from the infected animals' muscles. Then we were thinking about genetic characterization, and we could find out that European Union Reference Laboratory for Parasite recommends a multiplex PCR for the differentiation of different species. So here are the different species, and these are the targets which were targeted in the multiplex PCR. And if there are, for example, I will tell you only one, that if it is Britovi, then we will get 127 base pair and 250 base pair, 53 base pair amplicon in the multiplex PCR. If it is Trichinella spiralis, then we will get only one amplicon at 173. So this is the recommendation of the European Union Reference Laboratory for Parasites, and we also followed the same technique, and we could demonstrate that multiplex PCR recommended by the European Union was very much effective in differentiating different species. So this is the multiplexing, and these are the individual primers which were used for amplification. And in this way, we could get the information of of the parasites that we recovered, and we are maintaining all these parasites at IVRI in mouse model, and we could find that all the species, only one isolate was Trichinella spiralis, and it was recovered from Dehradun. Other species are not coming closer to Trichinella spiralis. So far, we could not confirm or get it published because we need permission from DAHDF for publication, because these two species are new to India, and we have to get the permission, then only we can disclose the name. But one such species is found only in Africa, not in other countries. How we could find it in India is a matter of really great challenge, and also we are trying to find it out how it got to India. Then also in our laboratory, Dr. Hiraram is working on expression of recombinant protein for Trichinella diagnosis, because you see in meat inspection, the whole carcass can be condemned if it is found positive for Trichinella infection. But after the slaughter, the farmer or the animal owner will not be agreed to this proposition that this carcass is not suitable for human consumption, and there is no system in our country by which we can compensate the animal owner with some monetary uh, benefit or monetary gain, and a pre-slaughter pre-slaughter diagnosis will be easier, and before slaughter, these animals can be discarded. So Dr. Hiraram is working on two proteins. He has already expressed the serpine and P49 protein, and he has utilized in Western blot analysis and also in ELISA. One protein, recombinant protein, serpine, has been used in the ELISA model, indirect ELISA, and he infected piglets with the larvae, live larvae, and then he collected serum from these infected piglets and used this in the ELISA format, and that's why he has written true positive, and some of these true positive cases are also false negative, and they are true negative, but some are giving positive result and bringing down the specificity to 75%. So it is still long way to go. You are seeing here that these are the true positive, but one is coming below the cutoff line, and in case of true negative, known negative, some are coming above the cutoff value, and thus it needs further study to increase the specificity. He is working on these lines, and hopefully we will be able to do it in coming years. Now, at the end of my lecture, I like to talk about the effect of COVID-19 pandemic in the last 3 years, and it has got a negative impact on the other diseases because of diversion of the available research grant and attention. And this is the priority. When COVID-19 came, it took the center stage, and the funding for other researches was greatly reduced. Then the manpower of very trained manpower could not be utilized properly because of
Sir, you are not audible. Sir, sorry to interrupt you. Voice.
from. Oh,
Yeah. Now clear. Yes, it's clear.
how long it was? How long it was silent?
Just now, sir. Just
Okay.
now.
Now it is audible.
Yes, sir.
It is audible. So these are the effects of COVID-19 I was telling, because we are now post-COVID era, and it had got a very major setback for the research on other priority areas, and available fund was diverted, and the trained manpower could not be utilized. There was a mental health of the resources was also not at that level where they can take up uh, research areas, newer research areas. There was global panic also. So, but there was one positive thing also, that the general preventive measures, this was helpful in containment of other diseases also, like the use of masks. So, meat-borne parasitic zoonosis, if we consider, then I think Tinea Solium cystic sarcosis, Toxoplasmosis, and also Trichinellosis are going to take the center stage. Trichinella search should be more methodical, and more areas should be covered, especially the wild animals and the domestic animals should be checked thoroughly, and there should be a strict meat inspection procedure in our country to make all these meat-borne parasitic zoonosis away from the human infection. In our country, we don't have this type of strict meat inspection. It is a cursory bit of thing, and it is half-hearted attempt. So with this, I finish my lecture. Thank you very much for giving me this opportunity to share my views on meat-borne parasitic zoonosis.
Many participants are there, means more than 400 participants they have registered. But yeah, but now means morning around 200 participants were there. Now also it's there around 150 participants.
150.
Yes.
Good. I'm
So
lucky enough.
yes, sir.
I'm lucky enough to
Yeah,
reach them.
definitely.
And I don't know whether they have noted, I I have given my email ID and phone number for the trainees. If they are interested, they may contact me through the mobile number and the email ID for further queries. However, right now I am also present to give any clarification. Thank you very much, Dr. Vaidya.
Thank you. Thank you so much, sir, for very excellent talk and updated and recent information you have presented about mid-born these zoonotic diseases, mid-borne parasitic diseases you
आप
have
भेज नहीं कि आई
presented. Now
नहीं
I request the participants, if they have
वहाँ
any
गई थी
questions from the
वहाँ को
participants, they can ask 2-3 questions. I think, sir, you have covered all the aspects. So very clear to all. And
Another thing is that the participants are feeling hungry, like me.
no,
It
sir,
is
not like
already lunchtime,
that. Yeah.
so they should be cautioned that during the lunch they should.
enjoyed your lecture as usual when you had come to Bombay for Rao Memorial lecture also. We enjoyed that lecture. Two, 3 points I would like to share here. One question is, have you come across Cystic bovis in the last 5 years? Because, you know,
No.
we were examining the meat samples. Of course, I'm talking about from 2011 to 2017, before my retirement. We could not get a single case of Cystic bovis. That was one question. Have you come across any report?
Yeah, I remember the last time I saw Cystic bovis was when I was at Pantnagar, and one of the PhD students of Dr. Bhatia, Dr. B.A. Pandit from Srinagar, he was working on sarcosistis of cattle. So at that time, he used to bring the beef or the cattle meat pieces from a slaughterhouse. And at that time, I found Cystic bovis. After that, I have not seen. But Cystic cellulose is very common even today.
And one more thing is, when I had a discussion with some of the scientists working on toxoplasmosis, and I was under impression that once the cat is infected, the cat remains symptomless oocyst passer for the rest of the life. But then I came to know that you have to examine the cat feces only for 7 to 10 days. And if she is negative for Toxoplasma oocyst, then you can, bring her inside the house, and then she can be safely reared in the house.
Okay. On this front, I like to tell that it is not true that the cat once infected will remain infected for whole life. This is not true. Scientifically, it is not true. But one thing, after repeated exposure to the infection by a cat, they will again start shedding the oocyst because they do not develop any protective immunity against this parasite. The parasite remains limited to the intestine and they do not develop immunity which can prevent further infection. But this is not true like Cryptosporidium or some other infection where the thin walled oocyst will be existed and again the cycle will continue. In this case, it is not like that.
Okay, because the pet practitioners often give corticosteroids, and maybe extra intestinal stages revert to the.
Yes. That is very true. That is very true.
Yeah.
That is very true.
Yes.
If immunosuppressed, the cat may shed or the cat may even suffer from acute toxoplasmosis because of the multiplication of tachyzoids. So, acute toxoplasmosis in cat has also been reported by Dr. Dubey.
Oh, yes. And just the last sharing, and that is we have also reported Trichinella pseudospiralis.
Yes.
That can propagate only in birds. Other species of Trichinella do not propagate in birds, but we could do that. Of course, Dr. Deshpande was working on that, but I faintly recollect it was, I think, published in Current Science, that old journal of Current Science.
Yes.
Pseudospiralis, because they don't form cyst, just for information.
Pseudospiralis has been reported, but I would suggest, with giving honor to and all due respect to all the researchers, if it has been done by molecular technique, then it is worth what I can say review. But if it is not done only on the basis of morphology or just they have not formed the cyst till now, may not be a right decision to arrive at a species level identification.
Maybe I am talking about 1984 or 85.
Oh, then sir, it needs further evaluation.
Yeah, because we propagated in the birds and we could find the larvae in the birds. So that is why we concluded that it is pseudospiralis.
Okay, that might
Thank
be.
you very much. Thank you very much,
Thank
Dr.
you.
And nowadays,
Thank
nowadays in my retired life, I am now venturing into the kitchen. I am developing a hobby of cooking, and thank you for giving kitchen rules also.
Kitchen, please propagate this also, that don't taste the meat before it is properly cooked.
Yeah. Pleasure listening to you.
Thank you very much. Thank you very much.
Thank you.
Thank you.
Thank you very much, sir. This is the real scientific discussion. So only one thing is that one of our student, that is Dr. Digambar Jundre. So he also worked on trichinellosis, sir, and in PCR study he found the Trichinella spiralis, pseudospiralis. So,
then it is confirmed. Now the question is that where you found this pseudospiralis, in which meat? Which meat?
In pig.
Which
sir,
animal? Pig.
Domestic pigs. Yes.
So it has been confirmed by multiplex PCR.
Yeah, confirmed by PCR. Yeah.
Another, my suggestion, humble suggestion to Dr. Vaidya, that whenever you are going for PCR identification, also try to clone that particular, or you send that PCR product for sequencing.
Yes.
And this will be a double proof that
Definitely.
that PCR product is sequenced, and then you align it with the published sequences. That will be very nice.
Yes,
That
sir.
is very
Yeah.
nice.
Yes.
Yeah, that is required. Yes, sir. Yeah. Once again, thanks a lot, sir.
Thank
for your
you.
time.
Thank you very
Yeah.
much. Thank you
Valuable
very much.
information. Thank you. Thank you.
Thank you.
Thank you so much.
Thank you.